All right, thank you for being here today at my general examination. Special thank you to the committee for all the time today, as well as all the time you've invested into me over the last several years. And I'd also like to thank my wife, who's been basically single parenting for the last 2 months as I get ready for this. Today's talk is going to be called Physics-Informed Principles of Design for Polymer-Based Trauma Interventions. Broadly, we're going to break it up into 3 parts. The first part's going to look at an injectable hemostatic agent named PolySTAT that our lab developed several years ago, and its role in clot contraction and fibrin network mechanics. Let me just hide these meeting controls real fast. All right. And then we're going to look at that same injectable hemostatic agent's role in whole blood clots, and how it might change relative to different species like rats and pigs, for example. And lastly, we're going to dive into low volume resuscitants and some work I've been doing there. Please hold all questions till the end. What's going on here? Apologies. Lost my cursor. There we go. All right. So this first part is investigating PolySTAT's role in clot contraction and fibrin network mechanics, and it resulted in a publication. The citation is there. Trauma and uncontrolled hemorrhage is the leading cause of death, preventable death, excuse me, a leading cause. And it's the leading cause of preventable death in the working population worldwide. It accounts for about 1.5 million fatalities globally every year. It's not just a combat thing, it's also a leading cause of death in the civilian setting. And it's characterized by rapid lethality, where the difference between life and death is measured in minutes, not hours. Early hemorrhage prevention is critical. Estimates as high as 35% of all uncontrolled hemorrhage fatalities could be prevented with early intervention by bystanders that are trained. The solution clinically is damage control resuscitation, which has a lot of different aspects to it. But first, the 3 big steps are recognize that there's a massive bleed occurring, stop that bleed with whatever means necessary, usually pressure, tourniquets, things like that, and then replace it, replace the lost blood volume so that you can reperfuse the tissues and restore oxygen to the organs. The stop it piece is where our injectable hemostatic agent is going to play, and our low volume resuscitants are going to be in that green box later on in the talk. Hemostasis is the endogenous response to an injury in the body. Begins with a vascular spasm to reduce blood flow. Then there's platelet activation, those yellow things, and aggregation, where they form the platelet plug to seal off the damaged endothelial cells, preventing the loss of blood. And then secondary hemostasis kicks off with the clot cascade, forming that cargo net of a fibrin mesh network that captures all of those platelets and begins to contract and pull down and seal up the damaged wound site. And it ends with clot retraction and then eventual fibrinolysis. Trauma complicates this dramatically. This triangle is the lethal triad. And basically, it's 3 hyperthermia, acidosis, and coagulopathy, and they all feed off of themselves into a negative spiral that leads to death pretty rapidly. Coagulopathies, one of the most deadly ones is hyperfibrinolysis, where you basically have that fibrinolysis step of the clot cascade super amplified and spread throughout the body, preventing any clots from actually forming because they're immediately digested. And that's what PolySTAT, our injectable hemostatic agent, was designed to prevent, is that hyperfibrinolysis. It's a factor 13 inspired hemostat. Factor 13 is that covalent cross-linking step. It's kind of that last piece of the clot cascade. And basically, we use RAF polymerization, which I'll talk about more in a second, to build this polymer backbone and conjugate on these fibrin-binding peptides so that they interact with the fibrin mesh network. They make a denser clot, a stiffer clot, and that is more resistant to fibrinolysis. What we do in the PUN lab is we design polymers for trauma applications and drug delivery. We just talked a little bit about PolySTAT. We're going to talk a lot more about it in a second. And then we also do these polymers. We design them for low volume resuscitants. We utilize this living polymerization technique called RAFT, or reversible addition fragmentation chain transfer. And the mechanism is drawn out as it was drawn out by its designers in the late 1990s on the right there. But I like to think of it a little bit more simply in terms of Legos. Basically, RAFT allows me to have great control over building a very monodispersed set of polymers or Legos. So I can act as the chain transfer agent, the control mechanism, and ensure that I get exactly the same length, size, architecture of my Legos, or in this case, polymers. Now, we said that PolySTAT is an antifibrinolytic, and one of the ways we measure and quantify whether or not it is working in vitro is with a viscoelastic assay called ROTEM. And so you'll see these two projections from the ROTEM. The x-axis is time left to right in minutes, and the y-axis is basically the size of the clot in millimeters. And so as time goes on in this ROTEM assay, if the clot is being digested, you'll see it taper off, as you see on the left in the negative control. So we've dosed these samples with plasmin to increase digestion and fibrinolysis of the clots. But PolySTAT resists that tapering off. So that's how you know that it's working. Interestingly enough, if we run any other assay in ROTEM without plasmin, we don't see any difference. So there's no difference in the mechanical properties of PolySTAT-treated clots relative to negative controls, unless we have that fibrinolytic step in place. One other thing that was interesting, Trey Pachon, who did a lot of the work on this and translational work from rats to pigs, he started to observe this artifact. So I just told you that without plasmin present in ROTEM, PolySTAT and saline negative controls should look the same. And yet something strange was happening occasionally when he was running these ROTEM assays, where it looked like PolySTAT, without any plasmin present, was undergoing fibrinolysis. And so he ran another test called APTEM that should inhibit any fibrinolysis, and he was still getting that same fibrinolysis response. So this was in whole blood. So the next step was to remove platelets and see if he was still seeing it. And so he did that, and lo and behold, the effect disappeared. Diving into the literature, it turns out that this is actually a common thing in ROTEM. What happens in ROTEM is you've got this cup, which is the blue-green thing up there on the screen, and you have a pin in the center of the cup. You fill the cup with blood or plasma, and then you initiate with factors to clot. And as the clot forms, it sticks to the sides of the cup, and it sticks to the pin and resists the deformation as you move left and right. If it clots too hard, however, it'll pull all the way away from the cup wall, and it will drop in terms of the response. And so there'll be almost no response all of a sudden as it pulls away fully from the cup wall. And that's what was happening. So visually, if you look at whole blood control there on the left and PolySTAT treated on the right, when you clot it, you'll see some clot formation in the whole blood on the left, but you'll see a dramatic clot contraction in the PolySTAT treated blood. So PolySTAT is somehow causing these clots to contract faster and harder and to a greater extent. So we started looking at, what was causing that, what was the mechanism, and he did some platelet studies looking at if PolySTAT was activating platelets, if it was increasing aggregometry, and he looked at also some platelet force measurements, which we'll look at in a second. But another thought was if PolySTAT was somehow reinforcing the fibrin network and making it more robust, maybe it was enabling it to translate the contraction of the platelets better. So the force measurements on platelets are here. Basically, he just clotted whole blood on a rheometer and measured the normal force required to maintain the gap height. And what he discovered is that PolySTAT does increase force generation per platelet and the rate of force generation, but still no effect in ROTEM outside of lysis. I had several years of experience working with my mentor, Matthew Armstrong, on blood rheology, specifically LAOS rheology. Blood is a viscoelastic material, which means it has some liquid-like and some solid-like characteristics. And I knew that blood undergoes nonlinear deformation in different strain regimes, in the nonlinear viscoelastic regime, that can be pretty dramatically different than the linear regime. We'd already looked at how whole blood clots responded in terms of the platelet response. So now we wanted to just look at a purified fibrinogen system. So we created that and we clotted it. And I created a protocol on the instrument to build basically a oscillatory shear strain ramp to look at both the linear viscoelastic regime, this small amplitude oscillatory shear, and then eventually increase the amplitude to the large amplitude oscillatory shear and move into the nonlinear viscoelastic regime. This is just a plot of the storage modulus and loss modulus. Storage is your elastic, your solid-like response, and the loss modulus is your liquid-like response or viscous response. Generally, you see it in the linear regime, there shouldn't be any change. They stay constant. As you enter the nonlinear regime, fibrin responds in a strain stiffening manner to a peak, and then it begins to soften, strain soften, and eventually yield. Some literature showed that physiologically relevant strain regimes in trauma patients are actually in the 1 to 100+% strain range, which is actually the nonlinear viscoelastic regime, which is not measured in ROTEM. ROTEM is only measuring the linear viscoelastic regime, which was our first clue that there might be something that ROTEM was missing that is actually happening in the mechanical properties of this fibrin mesh network reinforced by PolySTAT. So we looked at the elastic modulus, that solid-like response. And in the lower strain amplitude and intermediate strain amplitude regime, we see an increase in the PolySTAT treated elastic modulus response, but it's not statistically significant. But when we move to the higher strain regime, further into that nonlinear regime at 100% strain amplitude, we now see a statistically significant increase in the storage modulus relative to the controls. So PolySTAT is at least increasing the stiffness of the clots. As we look at the LAOS modulus or the viscous response, we start to see something similar, but it's statistically significant earlier on. So there's some type of a dissipative effect in terms of the clots response occurring in the PolySTAT treated clots differently than in the negative controls. And we'll talk more about that in a little bit. But first, going back to some of the work by the Weissel group and other groups, we have a pretty good understanding in the literature of how the fibrin mesh network is created with these fibrin monomers, how different parts of the fibrin monomers, which are in the bottom left-hand corner of your screen, how they interact with each other via alpha A knob and B knob and A hole and B hole interactions, as you can see over here in the top right of your screen. And so that forms this gray bundle, which we're going to call the protofibril. And when multiple protofibrils interact with these light blue little alpha C regions of the fibrin monomer, they then form a protofibril bundle. And those protofibril bundles are what make up the overall kind of cargo net look of the fibrin mesh network. We also know from literature where different parts of that fibrin mesh network, different parts of the protofibrils and the protofibril bundles strain and break relative to specific strain regimes. So for example, in the linear viscoelastic regime, the only thing that's really affected is the protofibril bundle itself. The interprotofibril bundle stretching and straining doesn't occur until you get into the nonlinear viscoelastic regime. And you don't see any of the intraprotofibril yielding occur until excess of 100% strain. So if G prime is increasing stiffness, back to the platelet kind of argument, it's well reported in the literature also that platelets pull harder on a stiffer substrate. So if PolySTAT makes a stiffer fibrin mesh network, as evidenced by the increase in G prime, then the platelets should sense that and pull harder, which is the response that we are seeing. But we're not sure if that's the whole story. So we talked that we're going to come back to the viscous response, and we will in just a second. But first, a little bit about the series of physical processes. LAOS rheology is difficult for a lot of regions, but one of the most challenging aspects to LAOS rheology is interpreting its results into something that's physically meaningful in terms of what is actually changing in the microstructure. Simon Rogers at the University of Illinois developed this series of physical processes about almost 2 decades ago now. And basically what it allows you to do is look at how the microstructure is changing with respect to time within an oscillatory shear cycle, rather than just looking at the holistic average values throughout an entire cycle. We can see how it changes at each time point. And it also allows you to look at yielding differently than you might think of in terms of classical mechanics, where you would pull a piece of metal until it exceeds its yield point and snaps. This looks at yielding in terms of soft matter as a process of restructuring and yielding at the microstructural level. And they provide you with a metric to basically quantify how much yielding is occurring and how much reforming is occurring within the microstructure. So we use that framework to analyze the data, and we got our yielding metrics for each of the different treated clot samples. And we don't really see a difference in terms of yielding at all. But when we look at repair, where a more negative number on this phase angle velocity, this metric of yielding and repair, more negative, it means an increase in repair. And we see a statistically significant difference in terms of PolySTAT versus the controls. So that viscous response, that dissipative response, we believe is due to this repair mechanism. And what we think is happening up here on C, the top right-hand segment of this plot, you'll see those protofibrils, and you see the interprotofibral stretching occurring, and you see the PolySTAT interaction with the blue alpha C domains. And as it stretches, those alpha C domains are disrupted, but PolySTAT is there to basically enable new alpha C domains to catch each other and reform the connections, basically allowing it to repair the interprotofibral bundle. And without it, as you can see in the top portion with the pink and the blue, you basically get a greater yielding occurring. And so our conclusion is that PolySTAT increases the stiffness of the fibrin mesh network, and that enables the platelets to sense that and pull harder. And then PolySTAT also enables a reforming and restructuring of the microstructure as you increase strain, which redistributes stress throughout the framework. Next, we're going to apply that same kind of framework of LAOS Rheology and SPP framework to whole blood of different animal species. One of the challenges of this injectable hemostatic agent has been translating it from small animal in vivo studies to large animals. Trey and the previous members of the lab have done a whole lot of work on this, and across many years and 3 large in vivo animal model studies, large in terms of scale, not in terms of the size of the animal, we see reduction in total blood loss, reduction in rate of hemorrhage, and significant improvement in overall survival outcomes in the rats. But in the pigs, we see none of those successful outcomes. Now, Trey had spent a lot of time trying to understand why we weren't seeing efficacy in pigs, and actually spent an entire chapter in his thesis going through this with computational fluid dynamics, looking at the allometric scaling laws and the law of Laplace as we increase the vessel size, for example, from the rat's femoral artery to the aorta of the pig, we dramatically increase the radius of the vessel, and that has impacts on the wall tension and the shear rate experienced by the clots in vivo. And all of this sums up to basically a 10x increase in mechanical forces experienced in vivo as you move from rats to pigs. But all of the work in the previous chapter led me to believe that there could be impacts on the fibrin mesh network and the clot itself that also are going on. And so intrinsic to the clots, I looked at physiological differences between the animals. And it turns out there's quite a few. Rats have twice as many platelets, have significantly higher and more variable fibrinogen concentrations, thrombin concentrations, and plasmin concentrations than pigs. And so with all of that, I kind of view this as a combined intrinsic clot mechanic PolySTAT modulated response coupled with the extrinsic hemodynamic factors that Trey explored. And we think that the combination of those is producing this failure to translate. So now we're going to actually dig into the rheology and see if that's real. And what we had learned from the fibrinogen systems was that we generate an enormous amount of data when we do this, especially as we process it through SPP and get all of those instantaneous time points. And one of the things that a tool that's useful in large data analysis is some machine learning tools. And an undergrad that's been working with me, Alan, had some experience with it. So we started applying some unsupervised learning models to do non-dimensionality or dimensionality reduction. And then some supervised models to try to create some synthetic data sets to see if we can increase the size of our data sets and better understand what's going on. First, rheology. In the pigs, so this is elastic modulus again, but now whole blood clots for pigs. And I will preface all this by saying the biological replicates are small. We do not have a lot of samples yet, so this is very preliminary data. But in the swine, we see a big increase in G prime, elastic modulus, from the negative control, which is in the light gray, to the PolySTAT in the black. And then the other increase occurs in the polyscram. And so this will be a trend that we notice some overlap between the polyscram, which polyscram is basically a same polymer backbone as PolySTAT, but a scrambled peptide for the fibrin binding peptide that should interact with the fibrin mesh network. So it's just a control for the polymer, different than a negative control like saline. But we're seeing an increase in both of them. Also, interestingly, though, and it's a little bit lost because of the logarithmic scale on the x-axis, we notice that PolySTAT shifts the point at which we start to yield left. In other words, it's stiffening the clot in pigs, but it also appears to be making it slightly more brittle. This wasn't as noticeable until we looked at rats, though, where the opposite effect begins to play out. So same increase in G prime, but now we're shifting the point at which we begin to yield orders of magnitude to the right, making the rat clots much more resilient, whereas in the pigs, it was going the opposite direction. And there can be a lot of reasons for this, everything from differences in the total platelet count to the differences in the strength of the platelets and size of the platelets, but that's going to take more data and more exploration. So more on that later. One thing that was confusing, as I said, though, is this PolyScram, PolySTAT overlap. So across all the in vivo rat hemorrhagic shock models, at the top of these Kaplan-Meier survival plots, you'll see that the PolySTAT rats had 100% in the first two and then 80% survival in the third test. But the PolyScram produced 20% survival, highlighted in the bottom, the blue and the gray there. So dramatically different in vivo results, yet overlapping mechanical responses in the rheometer. So that's going to require some more exploration as well. One possible solution for this could be that PolyScram adheres to platelets. And so the interaction could be coming in terms of the PolyScram with the platelets instead of the fibrin mesh network. But we're definitely not sure about that yet. So LAOS results are underpowered. We need to get more biological replicates so that we have an actual properly powered statistical loadout. There are trends in terms of species differences that are becoming apparent, and we're going to apply some machine learning to try to tease those out more. So unsupervised and supervised learning are kind of the two arms of machine learning as a whole. And we're going to focus in on the unsupervised learning first, dimensionality reduction, specifically UMAP. So what this is it takes large data sets that are highly complex, high dimensionality, and it flattens them into two-dimensional space and color codes them. And what you're looking for is just differences in terms of how far away or how close it is to other color-coded portions of the plot. And all it's doing is just trying to help you see trends that actually physically exist, but that you may not be able to see in the data set due to the size of it. So the top column is pigs, and the bottom column is rats, or excuse me, rows. Top row is pigs, bottom row is rats. We have each column is increasing strain amplitude. So 2.5%, so that's linear viscoelastic, and then 25 and 100% strain, that's nonlinear. And on the rats, we'll start at the bottom left. We see in orange is PolySTAT, blue is PolyScram, and green is negative control. And you start to see groupings of the orange away from the blue and green, where PolySTAT is starting to show a difference response, even in that linear regime, that the machine learning algorithm is able to pick up on that we don't see. Because remember, we don't really see the difference until we get into the nonlinear regime. As we move right and increase strain amplitude in the rats, now the PolyScram and PolySTAT start to get closer to each other. So you see the blue and the orange shifting left and coming closer together. As we get to 100% strain, we start to overlap more, although there's still some differences in terms of PolySTAT and the negative control, but we start to see the yielding. We're picking up on the yielding signature sooner in the machine learning dimensionality reduction than we are visually with the rheological data. In the pigs, there's more overlap earlier between PolySTAT and PolyScram, but there is still difference between the negative control, consistent with what we've seen in the rheology data. So primary determinant of the nonlinear clot mechanics seems to be species, and then a subordinate thing to that would be the treatment. And there appears to be this overlap that we observed in the rheological data between PolySTAT and PolyScram. And it does disappear as we get into the yielding regime. Next, we're going to move into supervised learning. And so this is where we built, Alan built off of the work of Safa Jamali, who's collaborated with Matthew Armstrong, my old mentor, out of Northeastern. He's basically developed a whole suite of physics-informed neural nets focused on rheology that are basically governed by rheological constitutive equations. So in this example, we're going to look at the Maxwell constitutive equation that's displayed there on the screen. And it's going to be able to control the output of this model that's learning off of our data sets and basically eliminate all of the responses that don't fit this equation, this governing physics-informed equation. And then, unfortunately, the Maxwell equation is not able to capture nonlinear responses because it's only linear. But then we're going to basically put this back through two other neural nets, more at a higher fidelity, and they're going to learn off of the experimental data again. And they're basically going to develop a response that's consistent with experimental data. So what does that look like? At 10% strain under the low fidelity, we get a Lisa-Zhao-Bowditch plot of stress versus strain. That's red is synthetic output, black is experimental data, and it's really pretty consistent. When we move to the nonlinear regime, though, we see that basically the synthetic data doesn't change, it stays the same. And what we see, other than magnitude, of course, it's that same kind of ellipse, but we get nonlinear deformation in the experimental data. So not great. You got to put it back through the next levels of neural nets, and you then get pretty consistent agreement with synthetic and experimental. And where we've been going from this is Alan's been trying other constitutive equations as well as trying to anticipate and predict the biological output, the experimental data output that we're going to get as we test additional biological replicates. So basically, he tries to synthesize new data before we even run it, and then we see how well it agrees. We still have a lot of work to do on it, but it's a promising approach that could expand our total kind of data set and give us maybe some more insights that we had previously missed. All right, we're going to move into the last part of the talk here. See what we're doing on time. Low volume resuscitants. So hemorrhage, uncontrolled hemorrhage leads to hemorrhagic shock. We talked about the damage control resuscitations and the 3 steps: recognize, stop it, and replace it. We're going to focus on replace it for this last portion of the talk. Hemorrhagic shock: as you lose blood volume, you lose the ability to reperfuse oxygen to your tissues and your organs, and eventually your organs fail. When you hit class 3, class 4 hemorrhagic shock, which occurs about 1.5 to 2 liters of total blood volume gone, you have minutes to an hour to live. That's where that kind of golden hour idea comes from. And you need to replace those fluids, which is kind of the solution. The gold standard is whole blood. However, we're designing therapies for the pre-hospital environment and the austere environment, think battlefields, where whole blood is usually not readily available. So lots of logistical concerns there. Next available thing, which is commonly used in those settings, is crystalloids, which have a lot of problems. One, you have to infuse them at large volumes, which creates a large pressure gradient, which can rupture blood clots and cause rebleeds. Next, if you don't heat it to physiologically relevant temperatures, you can induce hypothermia, which is one of those legs of the lethal triad, so that's no good. And lastly, it can induce dilution coagulopathies, where you basically dilute the clotting factors and inhibit the ability to clot. So not great options. Colloids are kind of the next best thing. And organic colloids have been historically what have been explored, starting with albumin, which has had mixed to limited results, moving into Hexten, which had its heyday in the beginning of the GWAT, so early 2000s. This is a head-of-starch-based colloidal approach. However, there's several issues with Hexten. It's no longer recommended. First, it leads to acute kidney injury. And second, many of the initial academic studies had questionable integrity behind them with dramatic retractions. Now, on the synthetic colloidal front, the Mangino group has done a bunch of really impressive work with PEG-20K. They showed that they can basically infuse PEG-20K in as a low-volume colloidal resuscitant and basically recruit 12 to 18 times the volume that they put in, pulling water out of the interstitial spaces of the body into the vasculature. So really impressive resuscitative effect. You don't have to worry about heating it up because most of the fluids are already at physiological temperatures, and you can recover dramatic pressures and The major problem with PEG-20K, though, is coagulopathies. So it does induce a coagulopathy, and so that's a problem. And that's where our therapy comes in. We're trying to improve on their work and make better polymer-based low-volume resuscitants. So we use RAF to do that. Our general target is a 60% total blood volume loss. So this is a massive severe hemorrhage event. You would have minutes left to live, and we're trying to replace that loss of total blood volume with a 10% total blood volume replacement. And so the way that works, or excuse me, the polymer that has been published by the lab is this radiant star architecture of a polyglycerol monomethacrylate. And the general idea of how it works is you put a high concentration of it in the capillaries through intravenous injection, and that generates an osmotic pressure gradient that pulls fluid out of the cells into the interstitial spaces, out of those and into the capillaries, restoring that pressure. And it demonstrated an improved resuscitated profile over Hextend and no coagulopathies, unlike PEG. And we got some funding so we could pursue it, and that's kind of the focus of my last bit here. Now, transcapillary refill is governed by the Starling equation, which basically says that hydrostatic forces pushing out of the capillaries into the interstitial spaces are balanced by oncotic forces in blue, kind of driven by that colloidal osmotic pressure. In a normal, healthy human being, that's primarily albumin, okay? So we're trying to kind of replicate albumin. Now, positive net flux is filtration, fluid is leaving the capillaries, and negative is absorption, where fluid is coming back into the capillaries from the interstitial spaces. Trey has worked on this a lot, and so they developed these design principles throughout the process. Coagulation would be the first one. And remember, we said that the original Radiant Star GMMA that they produced, it showed no coagulopathies relative to a control. And so I'm going to show you a lot of ROTEM results, and on all of them, good is up towards the top because the saline control basically is kind of the optimum. That's the goal you're shooting for. When you put polymer into blood, generally you have some inhibitory effect on coagulation. We're trying to minimize that. Now, we had seen no issues with the previous ROTEM results. So what we did was we made a more strenuous ROTEM assay to further expand our safety profile in coagulation. So you're going to see this GMMA that I said had no coagulopathies start to have coagulopathies. It's not because it's not working, it's because we created a more strenuous assay. In terms of water coordination, we're going to quantify that in vitro with differential scanning calorimetry, basically how many water molecules are being coordinated by each monomer repeat on the polymer. And then we're going to verify that with another in vitro assay called an oncometer that's going to measure the colloidal osmotic pressure. And so if you recall back here from the Starling equation, that's your blue colloidal osmotic pressure is the blue pi c term there. And generally for that one, what we want to see is an increase in the number of water molecules coordinated and an increase in the slope on the oncometer data. For biodistribution, since we haven't done any biodistribution in vivo with these new polymers yet, we're going to base it largely on the assumption that a larger macromolecule will stay in circulation longer, and we're going to measure radius of gyration with small angle X-ray scattering. And then all of these will feed into effective dose. So you're going to see effective dose throughout the rest of this presentation. Basically, right now, our current treatment is dosed at almost 2.5 times higher than PEG. And so we're trying to bring that dose down for a lot of reasons. One, we want to reduce immune responses. Two, we want to reduce issues in terms of cost and translation. And just on the whole, we want a more efficient polymer that we have to put less mass in and get a better response, ideally outperforming PEG. Our variables, the knobs that we're going to turn to try to get after these design principles, are these 5 things here. So backbone chemistry. So we're going to start with methacrylate chemistry that we've already looked at. We're going to move to methacrylate chemistry and eventually at the bottom, acrylamides. And so that's your increasing hydrophilicity as you go down that list. Then we're going to look at architecture going from linear to radiant star to bottle brushes. We're going to explore arm density. And we're going to play around with the sidechain chemistry of the acrylamides to see how sidechain chemistry changes things. And lastly, size. This is going to be the map for the rest of the talk. And I look at each of these different colored boxes as checkpoints along the way. So we'll start out with the linear sidechain study. And just to highlight, we are changing the backbone chemistry from what we have used, which is methacrylates, and we're going to move down to acrylamides. And we're going to just look at acrylamide sidechain differences so that the backbone chemistry stays the same. We built this library, or I built this library of polymers, and basically we changed up the number of hydroxyls and we added in a few Zwitter ions, and we're going to see how all of our design principles change. So this is the differential scanning calorimetry results, the water coordination. Larger bars mean more water coordination, and generally what we see is PEG on the left is low relative to our acrylamides, and then the Zwitter ions do significantly better, which makes sense. They should. Transcapillary refill, as recalled, is governed by this colloidal osmotic pressure term, and that's what we're kind of aiming for. So in the oncometry data, we want to see a larger slope, more positive slope on this oncometer data to indicate better water coordination in vivo. So we've got the same library of polymers basically run at different concentration gradients so we can get a COP profile. And you'll see that the Zwitter ions at the top, CBAA1 and TMAO, are kind of the largest in terms of water coordination in the oncometer. And then you've got the 2-hydroxyl is DHPA, and then PEG20K, and then HIA, which is a single hydroxyl, Sarinol, acrylamide, LGMMA. So the general trend, tris is at the very bottom, and that's a 3-hydroxyl polymer. The general trend appears to be decreasing the number of hydroxyls on the side chain, increases the COP response, and the Zwitter ions do the best. But this is on a molar concentration basis, and to go more clinically relevant, we're going to look at a mass concentration basis, and the trends start to change here. This is largely due to the molecular weight of the polymers, but also their efficacy in terms of water coordination. So PEG quickly jumps to the top because it's so small and it has the ability to recruit so much water. The TMAO is still doing well, but now acrylamide is closer to PEG, and we see that same trend of reducing hydroxyls increases the response. PEG and 20K are no good, though, because PEG has coagulopathic issues, and acrylamides are toxic, can't be used. So kind of where do we go from here? The next step is to look at effective dose. And so before I show you effective dose, I want to show you how we calculated them. We took Trey's in vivo work and the rest of the lab's in vivo work. We said, okay, PEG20K dosed at 10 mg per mil at 10% total blood volume produces effective survival in vivo. So then I took each of these polymers, I ran them on the oncometer, building these oncometer profiles, and I said, at 10 mg per mil, what's my COP? And it turns out it's 21.2, so that's where we got that number from. And then we took any other polymer that we have a profile on the COP for, we did a linear best fit, and then we combined that linear best fit and the COP kind of goal with some assumptions about the rats that we're going to test it in, and we can calculate dose from there. And so remember, we're trying to reduce dose with Radiant Star GMMA, kind of our gold standard at the top. 2.5 times below that is PEG, where we're trying to get to, and we can see the acrylamides are reducing the dose required with the exception of that tris 3-hydroxyl polymer. Next, we're going to look at coagulation. Recall I said that the Radiant Star GMMA showed good results there. Now you're going to see it drop. So the black arrow is pointing to the GMMA. It's now showing that it's inhibiting coagulation because we want those bars to get all the way up to the negative control. But it's actually still outperforming the acrylamides, which are doing very poorly in coagulation, with the Zwitter ions doing the worst. So the next question was, this tris popped up as doing pretty well here in coagulation. So we asked, can we somehow protect the Zwitter ions, maintain the water recruiting capabilities, but minimize the coagulation effects? And so in terms of water coordination, we made these two 50-50 statistical copolymers of Zwitter ions and tris. Water coordination shows that the copolymers basically perform as well as their Zwitter ionic sisters. In terms of COP, we see similar results. The homopolymers still do better, but the copolymers are close. In effective dose, similar results, no big changes, no big decreases. And so then we get to coagulation, the real thing we're trying to solve with this protective effect. You'll notice CBAA1, the homopolymer, is much lower than CBAA1 tris, its statistical copolymer sister. So we are protecting it from coagulation effects. And we see the same trend with TMAO and TMAO tris. So there is a way to protect these. It does work, and adding more hydroxyls seems to do the trick. In summary, from the linear sidechain study, increasing water coordination as we move from methacrylates to acrylamides was valid, so that hydrophilicity kind of trend was good. However, the coagulation impact was so bad that it's got us thinking we need to go back and relook the backbone chemistry. These trends we already talked about and highlighted in terms of coagulation and COP results, and the copolymers can be protected. The zwitterons can be protected with a copolymer approach. So next, we're going to now look at varying the backbone chemistry from methacrylate to acrylate to acrylamides. And so to do that, we generated a small library, basically alter the backbone chemistry like I just said, and do one with one hydroxyl and one with two hydroxyls, and let's see what happens. In terms of water coordination, I'll back up. Acrylamides are all solid black. The gray dots are your acrylates, and the gray lines, diagonal lines, are your methacrylates. You'll notice the picture of the Eppendorf with the foam and the water. That's HEMA. I cannot get it to go into solution after an entire night on the vortexer. So it is not water soluble, and you'll see that picture instead of any data from here on out. In terms of water coordination, methacrylates are the worst. Acrylates outperform acrylamides and methacrylates slightly. In terms of COP data on a molar basis, we see in pink the acrylamides and in orange the acrylates. Black is your methacrylate. And the pink, the acrylamides, they still do better, but the acrylates are close. When we move to a mass basis, however, the trend collapses, and basically the acrylates and acrylamides perform almost equivalently. In terms of theoretical dose, similar. Acrylates and acrylamides reduce the required dose relative to GMMA. But in coagulation, this is where things really got interesting. So remember, higher, closer to the 1, the saline control is better. PEG 20K is way down at 40% of that. Linear GMMA is about 70%. And the acrylates, in terms of GMMA acrylate, is up at 80%. But the HEA is the true star, basically had no negative effect on coagulation, even in this more stringent assay where our radiant star that previously worked is now struggling. Lastly, we're going to look at biodistribution, and this was interesting as well. Radius of gyration measurements via SACS showed that the HEA acrylate is actually quite a bit smaller than its acrylamide cousin. Excuse me. Generally, as I said, we want larger molecules to stay in circulation longer. Now, we're not sure if that's going to correlate exactly, and we need to measure radius of hydration as well. And I also need to measure the acrylate GMMA variant to see if that's also consistent. But we have a way to overcome this size limitation, and we'll talk about that in the next section. So in summary, methacrylates, they're pretty much out according to our design principles. Acrylates and acrylamides are tied. However, the acrylates have an edge because coagulation is kind of the most important metric right now. Because if you're able to restore blood pressure but just cause them to bleed out, it doesn't do you any good. So acrylates are going to be kind of our down-selected backbone chemistry moving forward. Next, we're going to move into architecture, and we're going to be focusing in on biodistribution, specifically size. But we'll look at all the other design principles as well. Before I get into that, one of the things that we changed when we moved back to radiant stars and looked at them, the original GMMA radiant star that was developed had this macro core CTA at the center, which is a 5 kilodalton dead mass. It can't recruit water, and it results in poor polydispersity. So you can't verify, you can't control effectively that you only get 3 or 4 arm radiant stars, and what you get is a mixture of each. It's also extremely expensive to produce the macro core CTA. So reduced water coordination, increased dose requirement, and increased cost. We looked at a different synthesis route. And so that's where the small molecule cores came up. This allows you to, at a lower cost, produce exactly the number of arms you want, 3 or 4 arms, and without the large 5 kilodalton dead mass in the center of your radiant star. And so it improves your dispersity, it improves water coordination, reduces overall dose required. Since the original Radiant Star GMMA was a mixture of 3 and 4 arms, we created a library. We created a library of 3 and 4 arm Radiant Star, just methacrylate-based polymers. And we went for small arm length, medium arm length, and large arm length, DP 50, 100, and 200 per arm. Just so you can see the difference in terms of polydispersity, how well we can control that we only get 3 or 4 arm out. This is your gel permutation chromatography output of, in blue, the OG original Radiant Star GMMA that Trey and the team had produced. And notice how broad the peak is. Broad peaks indicate poor control. They mean that you do not have a monodisperse polymer, you have a polydisperse polymer. The narrower the peak, the more control you have, the more monodisperse the polymer. And you can also see the difference in terms of size. So the shorter arms comes out first, and then the larger arms come out last. So as we go from 3S to 3 medium to 3 large, the time goes like that. This is some of the data. So you can see PDI is a quantifiable metric of dispersity, and you can see that the short arm and the medium arm 3 arm polymers have much lower PDI than the original OG variant. However, as we get larger, longer arms, I'm cooking it longer, getting higher conversion, and I'm losing control. In the 4 arm variant, we see a similar trend, good control over short and medium arm lengths, poor control over large, but still better than the original macro core CTA 3 and 4 arm mixtures. All right, so first biodistribution, radius of gyration via Sachs. This was interesting, and I wasn't expecting it because I sillily did not look up the literature on this first. As we increase the number of arms, I assumed wrongly that you would increase the radius of gyration. The reverse is actually true. And this is backed up in the literature as well as with modeling as far out as like 18 arm radiant stars. The more you increase the arms, the more you collapse the radius of gyration. But we can increase the arm lengths, and that will increase radius of gyration within the 3 or 4 arm kind of subcategories. So we can also see where the OG Radiant Star plays out, which is literally right in the middle of the two. All right, so water coordination, there's honestly not much of a difference between any of them. Pretty similar results in terms of differential scanning calorimetry, so we'll move on to COP. Here we start to see a difference. In terms of molar concentration, we see the 4 arms on top with the OG kind of in the middle and the 3 arms on the bottom. We see a general trend of increasing arm length generates a higher slope, so a better COP response. But when we move to a mass-based concentration system, things change. 4 arm is still better. PEG is better than all of them. And the shorter arms are better than longer arms, so kind of a flip of the trend. And this is due, once again, to that molecular weight kind of trade-off. So in terms of dose reduction, we see a dose reduction, but it's not uniform across all the 3 and 4 arm radiant stars. So you'll see that poor polydispersity in the 3 arm large polymer results in a pretty poor reduction in dose. But on the short 4 arm polymer, we reduce required dose by 43%. So in summary, increasing the arm count increases water coordination and oncotic efficacy, and it reduces the radius of gyration. And there's a clear benefit to the small molecule cores for all those reasons. And we're going to have to balance the oncometer theoretical dose trend because it appears to favor more arms, shorter arms, which would basically drive our RG down. And so we're going to have to balance that by looking at some in vivo biodistribution and figure out what that Goldilocks zone is. The next steps is to take that optimum chemistry, backbone chemistry, the acrylate chemistry, put it into another radiant star library where we now make the radiant stars out of the acrylates, and then do a pilot shock bottle study in rats. And at the same time, we will do a biodistribution in the same study in rats, and then eventually translate to pigs. We're also planning a DOTA mouse biodistribution study. The plan here is our group has used DOTA labeling on polymers pretty effectively with ICP-MS to do biodistribution studies in mice. And it allows us to multiplex. We can put 5 different metals on the polymers, so 5 different polymers at one time in a mouse. Pretty efficient way to go about it. And then also other next steps is to go to more complex shock models. So after a controlled bleed, we'll move to an uncontrolled bleed and eventually up to large animals. That concludes my talk. Are there any questions?